time for after the nice dinner. So a few announcements for today. One important thing is 12:30. We'll have, right before lunch, we'll have a group picture. So someone from outreach from the Institute will come and we'll take a picture, and we will meet at the entrance of the building. That's where we will have the group picture. Tomorrow, so this is the last day that we are here at the ICM. Tomorrow we will continue at the Botanical Garden. Katerina will send you instructions how to get there. So we will start 10 a.m. Remember, please, to try to generate the reports because you will use that tomorrow, right? So each group needs to generate a report deriving from the discussion, and that will be used on top of your notes from past, present, and future. If you remember what I explained the first day, that is a long time ago. And remember when you leave today to leave your card. It's important. The entry card, because otherwise, they will tell us things from the reception. So I think it's all I wanted to say. And then, we will have Sonia that we will introduce today. Yeah. Thank you, Sonia.
Good morning, everyone. Do you hear me? Yeah. We are starting our last day of discussions. Talks, not last day of the colloquia. We still have many things to discuss. And the first talk, we have 3 talks today. The first talk will be given by Pedrós-Alió about marine microbiome through space and time. The second talk will be given by Samuel Chaffron. He will share with us his experience in the community network models in plankton systems ecology. And the last talk will be from Marta Cole about global ecological modeling to stimulate future trajectories of change in marine ecosystems. And then we will have lunch and then discussions as usual. So, yeah.
Hi everyone, I would like you to meet Carles Pedrós-Alió. He holds a PhD in bacteriology from the University of Wisconsin, Madison in USA. He's a research professor at the National Center for Biotechnology of the CSIC. He has worked here at the ICM 25 years, too much, and he's a member of the American Academy of Microbiology. His scientific interest is to understand the ecology and diversity of microorganisms using genomics and massive sequencing. He has published over 200 scientific papers and 3 outreach books. And Carles today is here with us because he's going to talk about, as you see, the marine microbiome through space and time. So I hope you had a good time. And Carles, it's your time when you want. Thank you.
Thank you, Judith. Actually, I was here 30 years, not 25, so I'm older than you think. And also, I have 5 outreach books. I'm very proud of this, so, it's one. Okay, at any rate, I'm very happy to be here because I was here 30 years. And I'm very thankful to the organizers for giving me a chance to come back to Barcelona and to the Institute, where I miss many of my friends and colleagues all the time since I'm in Madrid. Then if I can turn this on. Yes. Well, okay, doesn't matter. Okay, so then I'll try to give you a very general impression of the marine microbiome. What do we know about it? And especially all the many things that we still don't know and that are challenging projects for you guys, for your future career. You really have a lot of work to do to really understand the marine microbiome. So in the ocean, In the ocean, we have a lot of organisms we can see, like, oops. Yeah, I know that this is a problem with this. Okay, there we are. We have a lot of organisms that can be seen with the eye, with the naked eye, but then we have all these microorganisms that by definition cannot be seen with the naked eye. And each one of them has a genome, and each one of these genomes has maybe between 1,000 and 10,000 genes. So the possibility to find interesting things out there is really amazing. And then there are some things that we can do with the marine microbiome, and the first one is that we can count the organisms. So we can filter a known volume of seawater on a filter, we can stain it with a dye, and then it's relatively simple to go 1, 2, 3, 4, 5, 6, something very simple. Since we then know the size of the ocean, we can do a very simple mathematical multiplication and find out how many different microorganisms are there in the ocean. And the number is this one. It's 10 to the 29th. This number is just amazing. It's impossible to intuitively understand it. Remember that 10 to the 11th, about here, is the number of stars in our galaxy. So if you recall Carl Sagan saying billions and billions of stars, well, he was referring to this point here. Remember that the computer that you saw on, I think it was on Wednesday, this Super Mare Nostrum supercomputer has 10 to the 16 flops. So it's somewhere in here. And we still have all the zeros to go up to the number of microbes in the ocean. So the possibilities for interesting genes are just amazing. Another thing we can do is to measure some of the activities of these microbes. And for example, we can measure photosynthesis and respiration. This is a scheme of the marine food web. The organisms are organized by size. So we have fish larvae here. We would have whales on the upper floor, copepods, ciliates, flagellates, bacteria, and viruses. And on this side, we have the phototrophic ones. And on this side, the heterotrophic one. So if we measure the photosynthesis of all these organisms, this is approximately 50% of the primary production in the planet. Remember that we had a talk on, I think it was on Monday, when we were told about this paper from 1999. So there will be many points of contact with previous talks in this one. And then if we measure the respiration of all the heterotrophic and autotrophic microorganisms, it turns out to be higher than 95% of the respiration in the ocean. So if we're interested in fluxes of gases with important effects in climate, like CO2 or oxygen, we should really look at the microorganisms. This is what the marine microbiome is doing. But it's doing more things. This is another function that is carried out by microbes in the ocean. Most of the algae in the plankton need to compensate the osmotic pressure due to the salt in the seawater, and many of them do this by synthesizing DMSP, dimethyl sulfonopropionate, which is a compound that is compatible solute and allows them to be happy in the ocean. But when they die or sometimes something happens, this DMSP is excreted into the water, and there it's reduced to DMS, dimethyl sulfide. So this is just a sulfur atom with 2 methyl groups. So it's something very volatile, very small and volatile. And so it goes to the atmosphere where it's oxidized to sulfate and sulfonate aerosol. And this aerosol acts as cloud condensation nuclei. So if we have a lot of plankton and we produce a lot of DMS and a lot of sulfate and sulfonate and a lot of cloud condensation nuclei, what we do is we increase the size and the density of the clouds, and therefore the albedo of the Earth. Therefore, solar radiation will be reflected back, and the ocean and the planet will be sort of cooled in a way. Now, if there is less solar radiation getting to the surface, maybe primary production will go down, and then we will have less DMS and less cloud condensation nuclei, and therefore less clouds, and the albedo will go down. So here we have a feedback mechanism where microbes, with the marine microbiome, is responsible for maintaining to a certain point the temperature of the planet, which is pretty amazing. There are more things that the marine microbiome does. This one, you already had a whole lecture on this on, I think it was Monday also, on the biological carbon pump. So I won't repeat what was explained very nicely, but I'll just remark that this part here is completely microbial, but this part is not. It's due to larger organisms. So here the marine microbiome is not the only responsible or thing to do this. And here in the decomposition, we have the marine microbiome acting again. So as you can see, there are processes that are happen at a very large scale that have implications for the whole planet, which are carried out mostly by microorganisms. And this is something that I find really amazing. Some organisms, as you know, create these calcium carbonate plates, like the coccolithophores. They form huge blooms in the ocean, and when they die, the sediment, they go to the bottom, and after hundreds of thousands, perhaps millions of years, these sediments are converted into rocks, and you have something like this. So I'm sure you were ready to accept that the marine microbiome is acting on the atmosphere and on the planet today. But before I thought about this, I wasn't prepared to accept that the marine microbiome also had an influence on the geology of the planet, which is something to think about. Okay, the third thing we can try to do is to estimate how many species of microbes we have. And I want to point out a large difference between what we know about the organisms on land and the organisms in the ocean. What you have here is a map of the world with the number of plant species in every area. So you can see that, for example, in the Andes in Colombia, we have many different species of plants. In the Sahara Desert, we have very few species of plants. So we not only have a reasonable estimate of how many species there are, which is 300,000 approximately, but we know how many are there in every part of the planet. If we now turn to the primary producers in the ocean, can go away. There, all we have is chlorophyll. So here you can see chlorophyll in land, and here you have the detail of the number of species. But here you have the chlorophyll, but nothing more. So the difference in our knowledge between land and ocean is really huge. So as I was saying, you guys have a lot of stuff to work on along your careers. Now, this is a slide that I stole from Ramiro, but I'm sure he has a more updated version, so I didn't put his name, so he's not ashamed of the old-fashioned slide. It's just to show you that lately we've had a lot of advances in the sequencing potential with this massive parallel sequencing. I still remember when Craig Venter was presenting his first results on the GOES expedition, and actually the first study that appeared in Science in 2004, and he would look at you intently and saying, I'm talking about massive parallel sequencing. We were very impressed. And now everybody's doing it, so it's not so impressive anymore. And we also have this supercomputing, as we saw the other day. So we have very powerful tools to analyze the diversity of the organisms. And we've had many of these expeditions around the world because the ocean is so large that you really need a lot of sampling to really cover enough. This is the Craig Venter's expedition. First was this one around the world, and then he did some additional ones. And this is the number of sequences that were obtained and the 6 million novel genes and so on. This was done shortly after the International Census of Marine Microbes. This was the first application of 454 pariosequencing to diversity in the oceans, and it was done simultaneously with the same techniques, the same bioinformatic procedure and everything, all these points you see. So there was another big advance in this sense, again, a lot of samples and so on. Then is the well-known Tara Oceans. You've heard a lot about this cruise already. Again, this is probably old-fashioned. Probably there's a lot more information now. And finally, Malaspina, that was done on the Spanish ship, the Hesperides. Again, there were a lot of samples and the sequences. So we have powerful sequencing techniques, powerful bioinformatics, and a lot of sampling. So we're in a position, supposedly, to really determine what is the diversity of the microbes in the ocean. This shows you the stations of Tara Oceans is this one, and the blue one is Malaspina, and the red ones is the census of microbial life, and well, here is Blanes Bay, which is also, of course, very important compared to the other sequences. And counting all of this, we came to these estimates. The Census of Marine Life at the surface found 65,000 OTUs. At the bottom, 10,000, so less OTUs. And in Tara Oceans, there were like 35,000 at the surface, and almost 4,000 at the bottom. These numbers, again, are old-fashioned, but it's just to show you that the two studies are not so different from each other. So we would think that, okay, we don't have the right number, the exact number, but we have the order of magnitude. But no, we don't. It's wrong. Because something happens. This is a rank abundance curve. In this axis, we have the number of individuals of each species. And in this one, we have the species ordered by rank. So the most abundant species is here, is number 1, and this is its number of individuals. The second one, the third one, and so on, down to the rarest taxa. Now, with some of the early molecular techniques, such as fingerprinting, you could get maybe 30 taxa in each sample, which is, I mean, at that time, this was great. With cloning and sequencing, if you did a very deep clone library, you could get up to 500 taxa. On the Gauss expedition with metagenomics, it went up to 10,000, and with this high-throughput sequencing mechanisms, you can go to many more. However, we still don't know how far we are in this part of the curve. So we don't know how many organisms are there that are escaping our techniques. This is a study done by Luca et al., in which they looked at many different studies, as you see, 300, 400 studies, 500 studies, and the millions of OTUs that were being discovered. And as you can see, this is a classical accumulation curve, and it was getting here to maybe 700,000 taxa of bacteria. So this is a lot more than what we found with Taraoceans and ICOM. And here is the same thing for archaea, and there are a lot less archaea. So this seems clear. The diversity of archaea is much lower than the diversity of bacteria. But as you can see, if we accumulate studies, now we find many more taxa that we thought we had with these big expeditions. And again, something, well, the estimate from these curves would be between 2 and 4 million prokaryotic OTUs. So we're quite far from a few tens of thousands. And then something that I understand happens. And since I don't understand it, I hope that you guys this afternoon in the discussion bring light to me, illuminate me, and so I can understand this. This is an example from clone libraries. And this is the number of clones that people were able to do. As you can see, these numbers today are ridiculous. But at the time, this was a big effort. And then in different studies, you could see these curves. Some of them were abating, trying to reach an asymptote. Some of them were not. And some of them seem to be increasing linearly, and so on. But with these different estimates of how far would this curve go, these are the numbers for 3 different estimates. So these are around 300, 100, 300, and so on. So the estimate was around a few hundred taxa. Okay? These indexes are supposed to estimate what is the total richness of your sample based on the beginning of the curve. So in principle, you should trust it. You say, well, yes, okay, there'll be a maximum 300 taxa. However, if you do this with a different technique, for example, in this case, it's 454, the numbers are completely different. Here, the numbers, look at this, 100,000, 200,000, 500,000 sequences. This cost me dearly because we had to use one complete 454 plate for one sample. So you cannot repeat this too often, obviously. But this is what we got. In a sample from the bottom of the Mediterranean Sea, the estimate was 5,000 taxa. This is what we found. In the surface sample, it was 1,600. And we could, you can see the 95 confidence interval is very nicely fit by the curve. And doing some statistics that a physicist did for me because I wasn't able to follow it very well, we estimated that we needed to sequence 4 million sequences, so we needed to go to 4 million in order to reach the 90% of the OTUs. So how come that with this technique we get to these very large numbers, and with the previous technique we didn't get those large numbers? There's a discrepancy here. So who can tell us that a new technique will not appear, a revolutionary sequencing technique, and then these numbers will be multiplied by reaching several orders of magnitude more? There's something weird there that I don't understand. Maybe something trivial, so it would be nice to discuss it in the afternoon. At any rate, there was recently an estimate by Losey and Lennon based on this question. If you have here you have the number of reads or total abundance of the different studies, and here you have the number of OTUs that were found. And this has this linear, it's not linear because it's log-log, but at any rate, it's a linear thing. And all these points here, these points are the points from many different studies. Some of them were Christmas bird counts, so they were done with a very simple technique, just watching with your binoculars or listening to the sounds of the birds. Some were done with sequencing techniques. So there are all sorts of studies of organisms very large and very small. And then they extrapolated. And now I'm sure all of you are saying, my goodness, they're extrapolating from 7 logs to 30 logs. How can you do this? You know, this is impossible. This cannot be done. My colleague, Susana Manrubia, asked for the numbers they had and repeated the calculations, and she found that, yes, this confidence interval was quite reasonable despite the very large number of logs that go in the extrapolation. But so if we at least provisionally trust these numbers, the human gut, this is the number of bacteria in the human gut, has 10 to the 6 OTUs, so 1 million. The global ocean has the 10 to the 29th that 10 to the 10th. And the Earth, the whole Earth, should have 10 to the 12th or to the 13th. So this is 10 to the 12 organisms in the ocean, which is, it's, again, amazing. I'm not sure if you believe it. I'm not sure myself I believe it. But these are numbers. So in order to say they're wrong, we have to find why. And this is the conclusion of this part of, one of the conclusions of this part on diversity. This is Robert May, a very famous ecologist. And 30 years ago, in 1992, in an ICM meeting here in Barcelona, he said the following. We have a catalog of all the celestial bodies that our instruments can detect in the universe. But we ignore with how many living beings do we share the planet. How about that? 30 years later, this is still true. So again, you guys have a lot of work to do. And then the question is there room for this many different species on the ocean? And the answer is probably yes. And once Ramiro takes his picture, and I'm going to show you a couple of examples. This is a study that was done in Vigo by Teira and colleagues. This was after the Prestige oil leakage. You probably don't remember it, but this was a very, very famous case of a tanker that liberated a lot of oil and so on. And the Spanish government realized they didn't have experts in oil degradation. So all of a sudden, there was a lot of money to do research on oil degradation. And of course, we all became immediately experts on the topic. For a while, we had money, so it was quite nice. At any rate, this is a very nice experiment. What they did was they used several mesocosms with water from the Ria de Vigo, and they added different amounts of oil similar to the Prestige oil and followed what happened. So here you have first days, 2 days, up to 8 days. This is the concentration of this oil. As you can see, there was some at the beginning, and it was degraded. And here, what you have is the abundance of this one organism, Cycloclasticus. It's undetectable in the water of the Ria with the techniques we have. You cannot detect it, as if it was not there. But here, it grew, and once the oil was finished, it disappeared again. So we have here an organism that wasn't there, at least to our visible techniques. Conditions changed. They were favorable for this organism. They grew. They actually performed a very important service to the ecosystem by degrading this offensive oil that was put there. And once the substrate disappeared, it also disappeared and went back to this unseen majority of microorganisms. And the nice thing is that if you add more oil, you have more of this bacterium. So here you have more oil, and you have more. And here you have more, and here you have more. So it's a very clear example of this organism being a specialist in degrading this compound. But you will say, okay, yes, but this is an artificial experiment. Nature is not like this, and so no, this is not going to happen in other places. So before I go to an example to convince you that it also happens in nature, I'll show you an example of our colleague Pierre Galland in France, who did an experiment where he could find an Archobacter, another bacterium, that was present in one cell in 10 liters of seawater, and it was able to grow and contribute significantly to ecosystem processes in his experiment. He actually put pieces of wood and look at the degradation of these pieces of wood. So this organism was in nature in this concentration, one cell in 10 liters of seawater. So our sequencing techniques are very fine, but we hardly ever sequence everything that is in 10 liters. So a lot of the bacteria and the microorganisms are outside our field of view. And to really show how rare can be, Pierre did this metaphor. If you have one bacterium that is equivalent to a human being, if you have one bacterium in one drop of seawater, it's like having a person in a football stadium. And one in 10 liters, like this Archobacter, is like having one person in the whole planet. So it's very rare, and yet, if conditions change, it's there. It has its genes, it has its abilities, can grow, and can process things in the ecosystem. So as I was saying, I'm going to show you now an example from nature, not an experiment. This was a cruise that was done in, as you can see, 2003, 2004, in the Amundsen, in the Canadian Arctic. The ship navigated to the Arctic, allowed the ocean to freeze around it, and so we could sample throughout the winter, which is something that usually is very complicated to do. And here, Laura Alonso, who is now in Asti, in Basque Country, noticed something. She was trying to do fish and marfish, and she noticed that in some of her experiments, she had this kind of bacteria. And for those of you who do not work for bacteria, these are the elephants of the bacteria. Usually they're tiny things, maybe like this. And these are very big, and also they form microgates. So she was amazed, what is this? What's happening here? So she started to do a lot of different things to try to see what was happening. And the first one is that she did clone libraries with the small bacteria going through a 3-micron filter. And you see you have alpha, delta, gamma, baruco, and this funny thing here, but it's not very important. But then in the large size fraction, this Xanthinobacterium is more than 3/4 of all the population. And this is, again, amazing, because beta proteobacteria, like Xanthinobacterium, usually are not abundant in the ocean. And yet here we had one that was making this bloom. So she did several things. For example, she looked at the sequences from these clone libraries, and all these LB numbers are her clones. And you can see that there are many other Xanthinobacterium from an alpine glacier, from Arctic cyanobacterial mat, from the Tibetan plateau. So they're mostly from very cold places. So at least the habitat makes sense, because the Arctic obviously is a very cold place. So here we have a bacterium that is able to appear in many different parts of the world as long as it's cold. Again, the environment has something that is useful for this one bacterium. She then developed a fish probe for this organism. And here you have the normal DAPI stain, and here's the probe, the positive probe. And counting the organisms, she could see that from January 24 to March 26, so 2 months, most of the other groups of bacteria remain about the same, but this Xanthinobacterium made a very nice bloom and then disappeared. And this is about 50% of the community. Amazing. We couldn't think of what happened, and the only thing that maybe was an explanation was that you have here temperature, salinity, and nitrate. At this time, there was an eddy that changed completely the profiles, the vertical profiles of everything that went through the area. So this eddy went through, and this bacterium found optimal conditions. It grew out of nothing, out of a very rare number, and became abundant and important in the ecosystem. And then it disappeared again. This is probably happening all the time, all over the ocean, constantly, in different ways, but we're just not aware of it because we're not there sampling every moment at every place. So again, you guys have a lot of things to look for in the future. And now we're going to show you something that I find spectacular. It's an animation that Guillem Salazar did when he was here in the Institute, working with data from Malaspina, so it's the blue line. And what you're going to see in the animation is in the upper part, you'll see a rank abundance curve for a station. And here you will see the Hesperides at that particular station. So here you will see the Hesperides moving along the track, and here you will see the ups and downs of different bacteria forming this rank abundance curve. So I hope this works, because, yes, there you are. So here's the Hesperides down here. And here you see there are some, it looks like the most abundant ones are constant there. Here there are some changes. And now in the Indian Ocean, look, there's a big change. There it is. And then it keeps going. And apparently these abundant bacteria are the same all over the ocean. But here we have a very long tail of rare bacteria that keep changing from one station to the other. Again, this is happening all the time in nature, in the marine microbiome, and we don't have the resources to really analyze it in detail. So we have 10 to the 29 cells, maybe 10 to the 12 species. And if you divide, each one of these 10 to the 12 species would still have this number of cells. So there's plenty of room, both in terms of appearance in time and in space, and in terms of numbers, for this very large number of species. And the thing is that, of course, they are not equally abundant. Some of them will have a lot of cells, and some of them will have very, very few, like this Archobacter or this Jantinobacterium. And the advantage of being rare is that if you, for example, the Jantinobacterium was abundant for a while, and then it died, or it disappeared, and it moved down the curve to the rare part that is undetectable. When you do this, there are several things that happens. One is that you cross a threshold for viral attack, because viruses move around by Brownian motion, and they have to find their sensitive cells by chance. And if the concentration of the susceptible cells is less than 10 to the 4 per milliliter, approximately, they don't find them. It's just a question of statistics. Of chance, they don't find them. So if you're very rare, you're protected from viral attack. And also, at some point, you cross this predator threshold. This is not as clear because some predators are not selective, and so they would eat everything. But most predators are not silly, and they prefer the large, active, juicy cells. So if you're rare and inactive, you're very likely small and not very attractive, not very palatable for the predators. So at least partially, you're also protected from predation. So this means that if you're rare, you can be there for a very long period of time. And now we come to the question of time. We've seen what we ignore about space, and now we'll see what we ignore about time. And I would like to place this in the context of our friend Mireia Mestre, that did this microbial conveyor belt idea. In this model, we have when a bacterium, for example, the Cycloclasticus or the Xantinobacter, finds good conditions, it will grow and will become abundant. But then conditions change and it will enter dormancy and slowly die and be diluted until conditions change again and come back to this other situation. So this is this conveyor belt of species becoming active and inactive through time. And the most clear example is the seasonal study. And here's a study of Adrià Oladell that he did in Blanes Bay. And I've put this, those who know the paper probably will confuse because I put it horizontally because otherwise it didn't fit. But here what you have is different years. So this is one year, 2, 3, 4, 5, 6, 7, up to 11 years. And every vertical column is an OTU. And as you can see, there is a periodicity. So these OTUs are abundant at this time of the year, but they disappear this time of the year. So for them, the conveyor belt goes around once per year. And this is very clear and very well known and so on. But my question was, if you have these bacteria that become abundant at one time of the year, then they become rare, and they become abundant again, how long can it be that these bacteria that were here doing nothing, not growing, can still go back and be active? So will they be dead after, I don't know, 10 years or 100 or 1,000 years? So the idea was, how long may it take bacteria to go back to this situation in the present? And in a way, this is a time travel. Because, 1 year is not very impressive, but 1,000,000 years is pretty impressive. And these bacteria are able to do that. Of course, they don't have this kind of machine, fortunately, because it doesn't work. But they have these machines. One is amber, another one is rock salt, another one is ice. I don't have time to go through the three of them, so I'll just concentrate on ice, but just let me say a couple of things about amber and salt. What you need for a good time machine are a couple of things. The first one is that the organisms have to be preserved alive. So it cannot be a very destructive place or anything like that. And the second thing is that there has to be a natural mechanism so that these bacteria can go back to the active biosphere and become active. Then amber is pretty tough. So it's hard to imagine a mechanism that would break the amber and release the bacteria. So it's probably not a very good time travel machine. And the other thing is that the total amount of amber on the Earth is very limited. So probably it's not very important. Also, there's only one lab that has retrieved live bacteria for amber. So it's difficult to believe. But then in rock salt, the situation is much better because there are little cavities with salty water left inside the rock salt crystals. And archaea, particularly salt-loving archaea, are able to persist in these little pockets for a long time. They have been isolated, in fact, from salt that was crystallized 280 million years ago, which is a lot. And for salt, it's easy to imagine mechanisms to come back to the present, because the salt rock can be raised to the surface, and then rain will dissolve it, and then the archaea will be released. There are many possibilities for salt. One of them is mining. If we mine, we collect the salt from the past, and we release the organisms in the present. And also, it's not only us. Elephants have been doing this kind of mining for also thousands of years. There are mines in Africa that have tunnels excavated by elephants of more than 2 kilometers long. And they move in the dark. They memorize the route, and they go there to scratch the walls and collect salt for their own purpose. So this has been going on also for some time. But the best, as I was saying, is ice. And the reason, well, there are many reasons. One of them is that ice is cold, and as we know, cold is a way of preserving bacteria. All of us have in the lab our freezers where we keep our strains for long periods of time. So nature has been doing this also for many millions of years. And the nice thing about ice is that it's inevitable that it will be eventually molten and the microorganisms will be happily released. And for example, this is an example from Rogers et al. Bacteria through birds, through particles in the air, clouds and so on, reach the headwaters of the glacier. They precipitate, they are frozen here, and the glacier slowly goes down to the ocean. It melts and it releases the bacteria again. And this, according to Rogers et al., may be 10 to the 50 years, which begins to be quite impressive. And further on, they estimate that, what am I doing wrong? Here. That between 10 to the 17th and 10 to the 21st microbes are released from ice every year. Of course, you recall that the cryosphere has many different types of ice. We have, for example, sea ice that melts and freezes every year. So here, again, we will have a 1-year cycle. But glaciers may have 10 to the 50 years. Permafrost may have millions of years. There was a snow earth period with also millions of years. So we have all sorts of time machines in ice that can take microorganisms from one place to the other. So, as I was saying, we had a snowball earth with this kind of period. We have permafrost, we have ice caps, we have glaciers, we have sea ice, and this is the rank abundance curve again. So the abundant organisms, through this blue arrow, may enter any one of these time machines. And many years later, it may be released through the orange arrows, and probably, of course, they will be rare. So they'll be joining this part of the core. And you can have several cycles. So a bacterium could go into snowball earth and then go to sea ice and then to a glacier, etc. So we have many possibilities for repeated time travels for the same organisms. And this is something that I think I can believe, because you have here a timeline. This is logarithmic. So here you have 1 million years, 10 million, 100 million. Here you have these studies I was telling you about from salt, or 280 million year old organisms isolated. And you have all these different studies isolating live organisms from salt. The yellow ones are from amber, and the blue ones are from ice. So of course it's hard to believe that this will be true. Maybe it's what's contaminated and so on. Again, I don't have time to go into the details of the techniques, but I trust very much this part of the core. And at some point you begin to think that maybe I don't believe it. But the question is, since this is up to the Proterozoic, and I was telling you a bacterium can enter several of these cycles. Actually, there is no reason why we couldn't go back up to the origin of life. If that were true, I know it's very fantastic and so on, or almost science fiction, but if that were true, potentially all the microbes that have lived on Earth since the origin of life could be contemporaneous and be present in this rare part of the biosphere. I realize this is going a bit too far, but I'm just mentioning that it's a possibility. And then this has implications for many things. For example, for this rare biosphere, because it has a 4-dimension. Up to now, we realize that bacteria are able to travel throughout the planet in migrating animals, through ocean currents, through the atmosphere circulation. They can move from one side to the other and eventually get to almost any part. But now we realize there's also a time aspect of this. So it's not only that they can travel from one part of the planet to another, but they can travel from the past to the present, thus enriching this rare biosphere with a lot of microorganisms. And therefore, maybe it's not that strange that we have this very large number of species, because extinction will be very rare, and speciation will occur at normal rates. And it has another aspect, which is the question of the pangenomes. As you remember, different strains of the same species share a core of genes. All of them have, but each strain has some genes that are unique to that strain and that allow it to adapt to particular ecosystems. But now the pangenome also has a time component, because these bacteria from the past might interact with the bacteria from the present and exchange genetic material. This has implications that I find hard to imagine and so on. But it has one implication for pathogens which is particularly worrisome. Because, for example, the flu epidemics of 1918, that was really killed, I don't know how many million people, but it was much worse than the coronavirus one. And this virus disappeared, but it has been recently resynthesized in the lab, and it was extremely virulent with mice. And the question is, after that epidemic, humanity developed resistance to this virus, but after so many generations, that resistance is not there anymore. So potentially these pathogens from the past can come back and cause another epidemic. And at the smallest scale, this has already been checked with reindeer in Siberia. There were some massive anthrax episodes, and the reindeer were buried in the permafrost. And now with global warming, this permafrost is melting, and the corpses are duly coming up to the surface. And there have been, again, epidemics of anthrax. So this is a real possibility that we have to consider. But to finish, this was my first Antarctic cruise in 1994. And there is this tradition of the last day of the cruise. You celebrate, there's been a lot of work, but you're very happy with the work you did. And of course, you take a whiskey on the rocks, and the rocks are pieces of iceberg. And the icebergs are not like our ice cubes in the freezer. They are not a solid block of ice. They have little bubbles of air. And that is the reason that we can reconstruct the climate of the past, because this ice has these little bubbles. So when you put the ice cube, the ice pieces in there in the whiskey, as it melts, it makes a fizzle because the little bubbles are coming. And it's extremely nice. You know, you're surrounded by this wonderful landscape. You're happy with the cruise that you've done, and you're going to enjoy this whiskey, which is fantastic. But then at the time, I didn't realize that I realized I was drinking a whiskey with all ice, but I wasn't aware that I was also drinking an ethanol-fixed sample of an ancient microbiota. So, thanks.
Thanks, Carlos, for this amazing presentation. I don't know if you want to ask something to him. Yep.
I have a question. Maybe it's a silly thing, but I don't know. You said that if you are very rare, you are almost protected from viruses. So I wonder if we could estimate how much time has passed through the last time that this species was abundant or that crossed this abundance threshold by, I don't know, looking to viral traces in sequences or some stuff like that. I don't know if we can try to find out a proxy or...
There are a lot of implications that at least partially could be explored experimentally, and this is certainly one of those. I hadn't thought about that, but you're right. You could check about these CRISPR sequences and see if you can correlate that with the time that this species has not been abundant. And maybe you could even do some positive controls by making some bacteria rare or safe from viral attack and see how fast that changes. Unfortunately, I don't know enough about evolution and I don't know enough about modeling to do this, but I hope that somebody who has this knowledge thinks it's interesting and tries to find out.
Trying to understand the high diversity of the ocean, of the microbes in the ocean, I come up with three different possibilities. One is that they have very high speciation rates. The other is that they have very low extinction rates. And the third could be that they have low speciation rates, lower extinction rates, but they have had like 4 billion years to accumulate diversity. So which of those do you think is the most probable?
I'll wait till your next Nature paper to find out.
Well, one thing that have these microbes is they don't have sex. And then the species that don't have sex, they have more possibility to be for a long period or one single cell and then come back again in life. If you have S, you have more difficulties because you have to find another, and sometimes it's not so easy to find the other. So you need at least two, and then you have to find each other. So this, I think, is the difference between these microbes and other on the organisms that they have sex. Then they accumulate much more diversity and they will save the planet because they can reconstruct again on the life if we go away.
Yeah, I mean, definitely. Of course, I couldn't explain the whole story about the rare biosphere, but one of the essential things is that bacteria do not need sex to grow. So potentially one single live cell could just fill the earth with its clones very easily and very, very quickly. And on the other hand, organisms that need sex, as you said, they need to encounter a member of the opposite sex, and this has some probabilities. And if the density of the species is very low, the chances of finding each other is very, very low. I have a very nice example with a bird from Hawaii, the pouli. It was discovered in the 1960s, I believe. And every year there were less birds. There was a point that there were only 3. And they tried to collect one of the males and bring it to to collect a female and bring her to the territory of a male, because of course the males hold their own territory, but the females are the ones that move around. And so they did it, and I mean, imagine that handling one of the only 3 individuals of a species is a tremendous responsibility. So people were really worried and very careful and everything. So they brought this bird to the territory of one of the males, and they instrumented the bird, and next morning this bird just went back to their own territory. So there was no, they didn't like each other. Then they finally captured this bird, and they realized it was not a female, it was a male. So it made sense that he didn't like the territory of the other one, and he ran away. And he had an infectious disease, and he was missing an eye, and he was so old that he probably couldn't reproduce anyway. So this species obviously was destined to extinction, and that will never happen with a bacterium. So I mean, your argument is perfectly true.
Yeah. As you mentioned also, if we compare the diversity with the Earth ecosystems and terrestrial ecosystem, we know that the diversity of microbes in the global ocean is much lower than the diversity on the Earth, in terrestrial ecosystem. Considering that we have less space and probably less ecological niches, how do you explain that?
I'm not sure I understood. You said that the diversity on land is higher than on the ocean? But that's for what? For animals, or?
Oh, even for protists or for bacteria, we know that much more diversity on Earth than in the global ocean.
But
much more.
How have you estimated that?
By molecular surveys that have been done in terrestrial ecosystems, we know that.
I don't know. I think
with comparable methods, actually, we know that terrestrial ecosystem are much more diverse.
I think we don't know enough, because I was showing with the estimates from Tara Oceans and ICM. And then different estimates were several orders of magnitude higher, and I think the same problems happen on land. So my impression is that we don't have an idea of the order of magnitude neither on land nor on the ocean. But maybe we can talk about this in more detail later.
Hi Carlos, thanks for the talk. I was wondering whether you've explored the distribution of functions along the rare biosphere, and is it structured by environmental conditions or stuff like that? Like the distribution of the main metabolic pathway, for example?
You mean in the rare organisms?
Yeah.
Well, in theory, everything is there. In theory, because if they are able to move around the Earth. Of course, an organism from the Arctic will be very rare in Antarctica, but the function will still be there, so if conditions are adequate, it will still be able to grow. I'm not sure whether that's what you wanted to ask.
Yeah, I'm thinking about, this structuration that you saw, like in the Salazar GIF, when you see the turnover within the rare biosphere. Could it be explained by selection of some functions by environmental conditions?
Yeah, of course. I mean, I think both the example of Psychoclasticus and Xantinobacterium show that the conditions change, and the organism that has the best adaptation to that one particular source of carbon grows and does what it has to do.
But how can we explain that also like super rare species also contribute highly to ecosystem functioning without high abundance?
No, but first you have to become abundant.
Yeah.
And then you have an impact, and then you disappear again.
Okay,
of course,
so
there's some...
In the example you showed, you said it highly contributes to ecosystem functioning when it becomes abundant? Because there's also like a review from Alexandre Jousset that say that some microbes are able to carry functions whilst remaining rare.
In this case, there are several papers like that. Yeah. In this case, you have to see what do they mean by rare. Because I'm talking about 1 in 10 liters. And that, of course, for sure, it doesn't have any function. However, there are many bacteria, for example, nitrogen fixers are not very abundant. They're sort of intermediate, and yet they have a very important function in the ecosystem. So, there's a whole range of possibilities in that case. But of course, I'm talking about really rare, and it's hard to imagine that they do anything significant.
And in the same review, they talk about, they talk about the complementarity of the rare ones also. Like they wouldn't necessarily perform functions, but like help the most dominant to carry out functions and help the efficiency of the system somehow.
Again, it depends how rare they are.
You are. Okay, okay.
Is this the paper by Pierre Galland? No?
No, it's from Alexandre Jousset and co.
Okay.
Yeah, it's from 2017.
Okay, so thanks again, Carles, for your talk. And now we can go to the other one. Thank you.